ahead and look at some examples of fret in action, various ways in which it can be used to answer biochemical questions. Things about whether molecules are close together, whether they're binding, whether something is inhibiting binding, whether the shape of a molecule is changing, all sorts of really cool things that you can do with fret. I'll show you how we can interpret fret data, as well as give you some ideas about setting up fret experiments. First, though, let's just review fret. So remember, fret stands for Förster, or Fluorescent Resonance Energy Transfer. Basic idea is you have two molecules that are fluorophores. So remember, fluorophore is something that can absorb light at one wavelength and then give off light at a different wavelength. What happens with fret is that the first molecule is going to give off light that has an energy that is what excites the second molecule. So remember, light's just little packets of energy called photons traveling in waves. And what can happen in fret is that if those molecules are close together, the second molecule can actually steal the energy, not as a wave, so you don't actually see the light given off by the first, because the energy gets transferred to the second molecule that then gives off its own light. And that own light it gives off is going to correspond to the emission wavelength. So the light that's given off by the second molecule is going to be the light that the second molecule normally gives off. But you're exciting that indirectly by exciting the first molecule. But you don't see the light that the first molecule is giving off, just the light that the second molecule is giving off, as long as those molecules are close together. Fret is super duper distance dependent. In fact, we can even consider it a sort of spectroscopic ruler, because there's this direct mathematical relationship between the distance between the molecules and the amount of fret that you see. So by measuring the fret efficiency, we're able to figure out the distance between the two fluorophores, the donor and the acceptor. If we put the donor on one molecule and the acceptor on the other molecule, what we have is intermolecular fret, so between two molecules. And then you can measure binding of those molecules. You can also put your acceptor and your donor on the same molecule, but like two ends of it, as we'll look at. And then depending on the conformation of the molecule that they're attached to, so often this is going to be like a protein. So maybe you stick a donor on one end and the acceptor on the other end, and then you can see, well, is the protein changing shape, bringing these farther or closer together. In the case of quench fluorescence, here it's a similar idea that you have one molecule, an acceptor, stealing the energy that the first molecule would have given off as light. But in this case, the acceptor doesn't give off light, it just steals the energy, so you don't see light if you excite the first. So in fret, you have two molecules. The first molecule, the donor, gets excited by one wavelength of light and lets off a wavelength of light that excites the second molecule, which gives off its own wavelength of light. And remember, with each of these transfers, you're kind of going down in energy. The process is fully efficient, so you're going to be losing energy. If you have lower energy, you're going to have lower frequency and a longer wavelength, so that you can still be traveling at the speed of light. If the molecules are far apart, if you shine light that excites the first molecule, you'll see the light that the first molecule gives off. But if they're close together, what you'll see if you shine light that excites the first molecule is you'll see the light of the second molecule. Assuming that those are like your typical fret, in the case of quench fluorescence, what you'll see is that if they're close together, you won't see any light if you excite the first one. And if they're far apart, you will see light. So it's kind of the opposite situation there, because with quench fluorescence, the second molecule doesn't give off light, it just takes it. So with normal fret, you want to look and see which wavelength light you're seeing. If you're seeing the wavelength from the first molecule, the emission of the first molecule, then they're going to be far apart. If they're seeing the emission of the second molecule, they're going to be close together. With quench fluorescence, if they're close together, you won't see any light if you shine light that excites the first. But if they're far apart, you will see the light that the first gives off. Let's go and look at some real examples. Most of the examples we're going to look at are going to be proteins, but there are also small molecule-based fret probes that can do cool things, like bind to various chemicals and change shape, and therefore change frets, and you can monitor things. So cool things, not enough time to go into, but I can provide an article if you want. Let's talk more about these kind of protein-based strategies, however. We'll see how there are multiple ways in which you can introduce a FRET probe. So you can either have one of those fluorescent proteins that you can put on the end of your protein. We'll see that. We'll also see examples where you add a chemical that will modify specific residues. So different ways in which you can introduce these sensors, and then you just have to think strategically about where to introduce these. So if you're putting a protein on, your kind of only options are going to be those ends, the N terminus and the C terminus. So what you can do is you can stick a donor on one end and an acceptor on the other end of the protein. And then if those termini are brought close together, then you'll get FRET. And so if you shine light that excites the first one, you'll see the light of the second one. But if they're far apart, you won't. And you can do this to measure conformational changes in the protein that happen for a variety of reasons, including sometimes the binding of a ligand. So here it can kind of act as a biosensor to tell you about the presence of a ligand based on the amount of FRET that you see. That would be an example of an intramolecular FRET where they're on the same molecule. We also can have intermolecular FRET where here they're on different molecules and therefore you can measure their interaction. In this case, so a protease, remember a protease cuts proteins, so this is going to be some sort of peptide or protein with a donor on one end and an acceptor on the other. And if it gets cleaved, then you won't see FRET. But if it does not get cleaved, you will see FRET. FRET can be used in a variety of different ways. If you use those like protein and sensors, so you use like your YFP, your CFP, so like your cyan and your yellow fluorescent proteins. You can genetically engineer proteins to have these on their ends, and then you can go and you can get cells to make those proteins and analyze the proteins in the cells themselves. There are also methods that we'll look at with single molecule FRETs or SM FRETs, where you have one thing immobilized on a slide or some other chip type of thing. Often this is used in combination with a method called tonal internal reflection spectroscopy or TIRF. Don't need to know the details, just know that it's possible for scientists to immobilize molecules onto slides and then look and see and measure fluorescence, because remember, you got to excite them and measure the emission, and you're able to do that in single molecule FRET, and you can do cool things like get kinetics. And you can get super duper fancy with having multiple colors. You can have multiple FRETs going on at the same time. You can have 3 color cascades, all sorts of really cool stuff with FRET. But let's just focus on some simpler examples. Starting with some in-cell stuff. So here's an example. Basically, they're trying to make a calcium biosensor, so to be able to detect if there's calcium present. What they did was they attached 2 fluorophores, so they have eCFP and eYFP. So on like one end, the N terminus, they can stick the CFP protein, and then on the other end, they can stick the YFP protein. Now what's going to happen is if these proteins are close together, you're going to get FRET, but if they're far apart, you're not going to get FRET. And between these two, there's this N13 region, and then this calmodulin, which is a protein that binds to calcium. What's going to happen is that when calcium is present, there's going to be a change in this M13 region that's going to bring the fluorophores close together. But if there's not calcium around, well, then they're going to be far apart. And so by measuring FRET, they're able to detect the levels of calcium, because the more calcium there is, the more FRET you should see. What they're showing here is basically they're showing the emission. And you can see that in the presence of calcium, you get this high FRET, you get this high emission at that 530, which is what is given off by the YFP. But without calcium present, well, now what you're basically getting is more of the emission of the first FRET partner. You're seeing this 475, but you're not seeing as much of the YFP. So this is allowing you then to detect how much of the calcium is present. You can also do cool things to use FRET to kind of like measure forces and based on distances. And you can do things like, this isn't FRET, but you can actually have like split GFP where you have 2 parts of the fluorescent protein, like the protein made in kind of like halves that then have to come together. various really cool strategies for using fluorescence and using FRET as biosensors. One example is you can have antibodies that are labeled with fluorophores. And if you have two antibodies that recognize something, such as two different antibodies that recognize one of the COVID proteins, this is like the nucleocapsid protein, NP, what's going to happen is basically when you have two antibodies with one's a donor and one's an acceptor, bind to that NP protein, they're going to be close enough together that you're going to get FRET. And so this can be used to detect the presence of the virus, as well as then to screen for things that prevent the virus from infecting cells. So very cool strategies that you can use, lots of strategies that you can use for FRET. So this is an example with using antibodies as a way to prevent having to actually label your protein itself. So this is actually a cool example where they're trying to measure not just are things interacting or where are things interacting. So remember, if you're in a cell, because you can have the fluorescence, you only get that FRET in the specific area where molecules are interacting. So you can actually use this to localize things within cells. But you can also use like fancy methods in order to try to study like dynamics and kinetics actually inside of cells. So in this example, what they're doing is they're actually like micro-injecting cells with one of the proteins. So they have one of the proteins is going to be expressed in the cell with a fluorescent protein fused to it. And then you have a second protein that's going to be injected into the cell. Now what's going to happen is they can start monitoring. So in the top panel, so this is going to be 0 seconds. So basically before they inject, then this is going to be them after they're injecting. So this is going to be 1 second. And this is going to be 30 seconds. So in the first panel, they're showing the emission of the donor. And so remember, you excite the donor and then it emits, and you can measure that. And in the second one, we're monitoring FRET. This is the donor and the acceptor. What would you expect to see if the two molecules are interacting? You'd expect to see a decrease in the emission of your donor and an increase in the emission of your acceptor. And you can see that that's actually what's happening over time, is that as you start interacting, you're getting a decrease in the donor channel and an increase in your FRET channel. And then this is just showing you kind of like what things look like in the absence of fluorescence. And I think they're showing the tip of their probe, which is kind of cool. I actually did something similar with like nanobiopsy type thing where I was actually trying to take things out of cells. Didn't work so well, but it was a great learning experience. So here's another example. Basically what they have is they have these two fluorescent proteins, ECFP and EYFP. And in between them, they have a couple things. They have this SH2 domain, which binds to a phosphorylated sequence. And then they have this sequence that it would bind to if that sequence was phosphorylated. So the substrate is going to get phosphorylated by the Src kinase. And this is going to cause it to bind to that SH2 domain. When they're bound in that way, well, now what's going to happen is that you are going to get the fluorophores to be far apart, and you're not going to get FRET. So if you were to go and you were to measure the light given off by CFP, you would find that you would mostly be seeing the light that was given off by CFP. And if you were to measure the light given off by YFP, which is going to be our acceptor, you're not going to see the light. So basically you've lost your FRET. And so this is what we can see with this wild type. If we have the phosphorylation, we lose FRET. It's a little confusing here because basically instead of showing FRET, they're showing CFP to YFP. But remember that this means that we're getting more of the donor emission than we are of the acceptor emission. So that's why it looks kind of backwards. But you can see that then if they add a phosphatase, what's going to happen, they remove that phosphorylation, and then these come together. You get the FRET, you're not seeing as much of your CFP. What would happen, they wanted to see if like you made mutations to the kinase. If they make mutations, well, now what's going to happen is that they're going to not get the phosphorylation, they're not going to get this conformational change, and therefore they're going to stay having this FRET. And so they're going to see low emission of their CFP, and that's why you see these lines here. So you can do a lot of really cool things to figure out information about proteins, about functioning. various cool things. You don't need to know all these details. These are just examples to kind of illustrate the types of things you can do with this. Inside of cells, we can localize things. We can study what's going on in like a real environment. But often we want to do things where we can control things better. We want to really get down to the nitty-gritties. We need more control. And inside of cells, it's hard to tell what molecules are we actually dealing with, how many. Don't get the kind of resolution that you might want. Another option is single molecule FRET, or SM FRET. Typically, you have things immobilized. You don't always have things immobilized, but you can have like a slide. You then attach a molecule to the slide, and that molecule is going to be labeled with the fluorophores. Or you can have one molecule that's labeled and then you flow other things over it to see if they interact. But often you're going to have things like immobilized with biotin and streptavidin. These are going to be really common. Here they have a nickel on here and then a his tag on the protein. We could use like nickel resin to purify proteins that have a his tag. We can also use like nickel coated slides in order to capture his tagged proteins. And then if these have the fluorophores on them, well, now you have a way where you have these single molecules immobilized, and you can then study how they are moving and interacting in this sort of things at a much greater resolution than you would be able to inside of cells. Let's look at an example of what some of this data might actually look like when in one of these single molecule experiments. So here they have this molecule with a donor on one end and an acceptor on the other end. And it's linked to this slide through biotin and streptavidin. What's going to happen is that when the molecule is in a conformation in which the donor and acceptor are close together, you're going to see FRET. And when they come apart, you're going to lose that FRET. Now, these molecules might be going back and forth and back and forth and back and forth. So you can actually measure their conformational dynamics. You can measure the switching from the FRET to the high FRET state to a low FRET state to a high FRET state, and see something like this, where you have on your y-axis, you have like the FRET. So we can think about our FRET efficiency, remember, is going to be dependent on that distance. And then we can graph the amount of FRET versus time in order to get an idea about how much time these molecules are spending in these different conformations. What you might also see is like a direct counts. So basically what they do is they take the counts. So basically how much fluorescence do they see from their donor from the donor emission channel or from the acceptor. And then what they can do is they can like graph the ratio of those, and then they get this like FRET signal that like we were looking at before. But what they originally get is something like this. So you might see something with counts. So in this example, basically what they have is that this green trace is going to correspond to the emission of the donor. And then the red is going to correspond to the emission of the acceptor. What's going to happen is that if they were not interacting, you would basically not see the acceptor fluorescence, but you would only see the donor fluorescence. And so when you have something like this, and then you look down, this is going to show you that you don't have FRET. FRET's not going on here. Don't worry about photo blinking or photo bleaching. Those are just like what happens when the fluorophores get tired or do weird stuff. But what you see when you have basically this really, really low amount of your acceptor emission and a really high amount of your donor emission, this is going to be an instance where you do not have FRET. So these are examples of places where you don't have FRET. So the molecules are far away. But then you have examples where they come together. And so what is characteristic of FRET is that you're going to see a decrease in the donor at the same time you see an increase in the acceptor. And so you're going to get something like this. And this then is going to correspond to high FRET. Note that in the last example, it was really nice. It was like we're going from like 0 to 1 or whatever. But things are typically more messy, which can be annoying, but it can also be helpful because not only are we seeing these different conformational changes, but we can actually detect that they're actually kind of different efficiency states. So they're kind of like, remember, because it has that distance dependency, if they're kind of an intermediate amount apart, you can actually detect that. So FRET can be used to detect different conformations. And then often what's going to happen is that they'll plot it on some sort of like histogram, which shows like the probability that it's in one state versus the other. So there are different ways that you'll see FRET data, and we'll look at some examples. So let's look at a couple examples that are both going to involve that spike protein, the coronavirus protein that sticks up off of the surface of the virus and allows it to latch on to cells. And it does that by binding to the ACE2 receptor. So ACE2 is a membrane protein in our cells that binds to the spike protein in the viral. And then this allows for a shape change that's going to then merge the virus with our cells and allow the virus to dump its stuff in. One of the strategies for combating the virus would be to prevent the binding of the spike protein to ACE2. And remember that the specific region that binds is this receptor binding domain. So what they're going to do is they're going to use FRET to measure the binding of the receptor binding domain to ACE2 in living cells. TR, this is time resolved. Don't worry about it. It's just a type of FRET. So what are they going to do in this experiment? Well, if they want to measure the interaction between the RBD and the ACE2 protein, what do they need to do? They need to label the two of them. And instead of sticking a GFP onto one of them or a YFP onto one of them, they want to use something smaller. They don't want to interfere with the actual protein's interactions. Because you imagine you're trying to detect how they would interact, and then you stick some big bulky thing in between them. That's going to change how they're interacting, and that wouldn't be very helpful. Instead, what they want to do is they were going to use one of those small molecules. So how are you going to attach a small molecule to a protein? Well, they actually take a pretty cool strategy. They put this thing called a snap tag on the end of ACE2. So they genetically encode to put a little snap tag on the end. Basically, it's this little tag, SNAP is an O6-alkyl guanine DNA alkyl transferase that catalyzes its own covalent binding to fluorescent derivatives of benzyl guanine, such as LUMI4-TB. Don't worry about the details. Basically, there's this little tag that then they have an enzyme that will specifically add a fluorescent molecule to it. So this is going to then allow them to label the ACE2 protein. In order to label D2, well, here basically what they did was they used lysine labeling. So lysine, remember what functional group do we have with lysine? We have an amine group, and the amine group is amenable to modifications. And so basically you can conjugate it with this D2 dye. So they're basically able to use that lysine to label the lysines with a dye. And this is a common strategy that's used to label various proteins, but it's not going to be as sequence specific. There are also ways that you can label cysteine. Again, these aren't details that you need to know. Just know that there are ways that you can fluorescently label proteins if you're working with them in vitro. So because they're basically going to be purifying this RBD, so the receptor binding domain, they can then modify it and then do their experiments. Also, they kind of have this unique situation with ACE2 because it's a membrane protein where they can basically do this reaction while the protein's embedded in the membrane. They can add the enzyme that'll do the modification and then kind of wash the enzyme away. So a cool strategy so that you can actually be dealing with living cells, but still modifying the protein. So now what they want to do is they want to detect the interaction and the loss of interaction if you have an inhibitor present. So we have a fluorophore on our ACE2, which is called this LUMI4-TB. And then we have a fluorophore on our receptor binding domain, our D2. What's going to happen is that if the receptor binding domain binds to ACE2, so binds to our cellular receptor, we're going to get FRET. So if we shine light that excites this LUMI4-TB, we should see the emission of our D2. But if they're not, only if they're interacting. If they're not, then if we shine light that excites LUMI4-TB, what's going to happen is we'll see the emission of that. And so this is what they end up doing. Here they're doing a binding assay with this. So here we're looking at one of those equilibrium binding graphs. We're looking at concentration of our x-axis, and then they have this FRET ratio on our y-axis. So the more FRET you have, the higher the value here. And what would give you more FRET? Is that indicating binding or not binding? Yeah, in this case, it's indicating binding. So they're showing that when you get binding, you get this higher fret. They're measuring like total binding and like non-specific and stuff. Basically, they have a control to test for non-specific binding, where they have a bunch of unlabeled stuff to see if a bunch of unlabeled RBD to see if they're still seeing fluorescence. And so they're seeing they don't get fluorescence if they've saturated their receptor with the RBD and all's good. And so then they say, okay, well, that's all basically, if you subtract that, we can consider that all this binding we're seeing is basically specific binding. Okay, so from a graph like this, we can go and we can determine the KD, right? And so what would our KD be in this case, approximately? Yeah, it's going to be about 15 or so. Basically, it looks like the peak is about like 2,500, 3,000 or so. Go somewhere over here, come down. Yeah, you're about 15 nanomolar or so. And so remember, this is like one of the reasons why it's kind of nice to have those log graphs, right? Because you can actually see the inflection point better and these points aren't all scrunched up. But anyway, this is what we have, and you can determine things like KD. Can we tell anything about the kinetics here? Can we tell about K on and K off? No, we can't. If we only have the KD, if we only have this equilibrium data, we can't tell about kinetics. But with their strategy, well, they are able to tell about kinetics. And so they can set up their experiments. So what's going to happen is that they can measure the actual binding. And so when you get this, you can see association. You can get your K on. And then if they then do a dissociation assay, they're then able to see the release over time and get the K off. So that's what they're showing in this figure. And note that here they're basically showing how they got the KD from the kinetic data versus how they got the KD from the equilibrium data. And so you can see that they're not exactly the same, but they're within like a few factors of one another. Okay, so now they want to go and they want to look and use this as a screen for inhibitors. So if an inhibitor is present, do we expect to see more fret or less fret? We would expect to see less fret because you're not going to have as much of the interaction there. So if you were to mix competitor and your labeled RBD, well, now what's going to happen is that the more competitor you have, the less labeled RBD that will be bound. So remember our Ki? That's going to tell us about the strength of the competitor or of the inhibitor binding. So when we talk about enzymes, we talk about Ki in terms of the inhibitor binding to the enzyme. When we talk about a competition binding assay, the Ki is going to correspond to the concentration that inhibits the binding of half of the other thing. So in this case, you have a labeled RBD bound, and then you introduce a competitor, so an unlabeled RBD for your control, or you have, in this case, this LCB1V3, which is going to be an unlabeled competitor. When you have no competitor, you're going to see all of it's going to be bound to the labeled RBD. Then as you add competitor, that labeled RBD is going to get pushed off, and so you're going to see a decrease in the signal. And you can then look and see how much it took to push it off in order to get the value of the affinity. And so if we look at these, we can see that if we comparing the unlabeled RBD to the labeled RBD, so that should tell you about kind of the affinity of the RBD. So it's kind of like your control, your comparison point. We can see that it's going to be, so we go to the halfway point, that's going to be somewhere around 10 to the minus 7. So that's going to be so remember that 6 would be micro, 9 would be nano. So we're in between those two. If we look at this LCB1V3, well, here we see that we're shifted to the left. This is indicating a higher affinity. It takes us less of this to compete off the RBD than it took the RBD to compete off the RBD. So this might be an effective way to kind of get to that binding site of ACE2 first. This is a cool one too. Well, in this case, what they're doing is they're looking at these things called nanobodies. llamas have these really cool, like tiny little antibodies that you can make these like nanobodies, which are then smaller, easier to get into cells and things, and kind of can wiggle into nooks and crannies. So that's the strategy that some people use to target all sorts of things. And so they were testing the nanobody binding, and that's what's shown here. Okay, so that was measuring binding. So there we had two molecules that was intermolecular. We had a fluorophore on one molecule that excited a fluorophore on another molecule. Now let's look at an example of intramolecular fret. We have two fluorophores on the same molecule. In this case, we're going to use it to study conformational dynamics. So that spike protein, remember, it's the RBD that's going to be binding to ACE2. And in order to do this, it kind of has to adopt this up conformation. So the spike protein, basically, it's a homotrimer, so it has these 3 copies of the same protein. Each of those copies is going to have one of these receptor binding domains, and it can be in the down conformation or it can be in the up conformation. And when it's in the up conformation, this is when it can bind to ACE2. And so scientists wanted to study how often it goes up and down and up and down and up and down and up and down. And so in order to do this, they were going to use fret. They're going to take advantage of the fact that if fluorophores are close together, they'll be able to see fret. And if they're far apart, they won't see fret. So by designing fluorophores, like locations, strategically, you can then monitor conformational changes in a protein. In this paper, that's what they did, and they wanted to see how specific variants of the protein affect things. So how do mutations that change the protein affect, or do they affect how often it's going up and down and up and down and all that stuff? So this is a really cool paper by Yang et al. That I thought I would use as an example. What they're going to be doing is they basically have these like lentivirus particles. So basically this is like a safe virus, but they put the instructions for making the spike protein. And they can then anchor this virus onto a slide. They use this like biotinylated lipid so that they can then attach it to streptavidin, which is attached to biotin on the slide. So they have a way to immobilize this virion on the slide surface, so they're able to measure things kind of like in a more natural environment, but still have that tight control. In order to be able to measure the dynamics, they're going to introduce 2 fluorophores. So how they're going to do this is basically, so in here, this red dot is going to be this like A4, this is one of their fluorophores, and this green dot is going to be this Q3, another one of their fluorophores. And so you can see that this is when all of them are down. Are these close together or far apart? Compared to over here, where one of them is open. Yeah, these are going to be closer together here. So are you going to see more fret if they're all down, or more fret if one is up? Yeah, you're going to see more fret if they're down. So you get higher fret if you are in the down conformation. Things can get really complicated if you were to have like all of them labeled. So they basically were very strategic and only labeled like a small amount of them and had most of it be unlabeled, so that they would on average have like one subunit that had both labels per spike protein. And therefore they would have the least amount of noise. As to how they actually did that labeling, it's pretty cool. And they kind of like put these really, really short little sequences inside of the protein sequence. So instead of putting them at the end, that wouldn't work for them because, well, the protein, they'd be like way too far apart. You wouldn't be able to see if you had fret or not. We need to choose something sites that are going to be close together, but then how are we going to get them labeled? To do this, basically what they did was they used these cool little like sequences that are recognized by these specific enzymes. So by like ACPS, as well as like transglutaminase and stuff. Basically, they have ways that they can then introduce specific labels in specific locations. And then by being careful about how much they use, they're then able to get on average one of the spike protomers, so one of the individual units labeled per spike protein. And this is going to then allow them to study the dynamics. And so what you see is you get something like this. So this is our like fluorescence. This is basically our counts. So you can measure the fluorescence of the donor and the fluorescence of the acceptor. So if you have a region like this whole region, like these regions, what is this showing us compared to this? Well, in this region, what we see is basically we're seeing mainly the emission of our donor and very little of our acceptor. So we're not seeing FRET here. And indeed, if we look down, you can see that there's not FRET. But if we look at some place like here, well, here we see the decrease in the donor, an increase in the acceptor. And if you look down here, yeah, we do have FRET occurring. And you can see that kind of like we saw before, you can have intermediate levels of FRET. So they talk about like open versus closed versus partially open. So they see these different populations. So what do you think would correspond to the open conformation up here? So remember that when they're fully open, those fluorophores are going to be far apart from one another. And so you're not going to see FRET. And so the open would correspond to things happening down here. What would correspond to the closed? Yeah, the closed would be up here where you see that high FRET because they're going to be close together. So what they wanted to do is basically look and see if the mutations were going to influence things. It's kind of confusing because basically D614, this is going to be the wild type. So this is our wild type. And this is D614G. But they write it G614, which is weird. But basically, this is going to be our mutant version. This is one of the earliest mutations that occurred and kind of really quickly took over. And so scientists were trying to figure out why. And one of the reasons they thought was that it might influence how that receptor binding is going up, down, because you can imagine if it needs to be in the up to bind to ACE2, if you had a mutation that made it up more often, this would increase its probability of binding and like being able to infect cells. And in fact, if we go and we look at that wild type, we can see that it spends a lot of time in each of these different states. It spends a lot of time in the closed state, not that much in the opened and not that much in this partially open. But what happens with the mutation is now you're spending more time in this partially open state. And this can make it so that it's then able to bind ACE2 more readily. What happens if you actually add ACE2? Well, if you add ACE2, well, now what's going to happen is you're going to stabilize that open state. And in fact, that's what they see. So both with the wild type and with their mutants, if they add ACE2, they see that they're stabilizing the open conformation. So again, does that open conformation correspond to low FRET or high FRET? Yeah, that's going to correspond to low FRET. So that would mean that you would have more of these instances where you would have things like this, where you're seeing mainly the excitation of the donor and not the acceptor. Hopefully the examples were helpful and kind of cool and not just overwhelming. Remember that you don't need to know all those details. They were just examples. And instead, what I want you to focus on is those concepts.